Welcome to the current knowledge on the potential for virus transmission through swine feed. My name is Dr. Cassie Jones, and I'm an associate professor in the Department of Animal Sciences and Industry at Kansas State University. Joining me in the development of this webinar include Jordan Gebhardt, current DVM PhD student in our program, Megan Niederwerder, who is a virologist and DVM PhD in the Diagnostic Medicine Pathobiology Department in the College of Veterinary Medicine, as well as Dr. Steve Dreetz and Dr. Jason Woodworth, members of the K-State Applied Swine Nutrition team. The date of this webinar is September 20th, 2018. As we consider viral transmission through feed, I want us to be familiar with three specific terms. The first is the reservoir. A reservoir is a habitat where an agent lives, grows, or multiplies. Bacteria and viruses can have reservoirs that include humans, animals, and even the environment or soil. The transmission, however, would be the method of transport from the reservoir to a susceptible host. Examples of transmission include direct contact, which are by far the most likely means of transport of viruses, such as by having droplets sprayed on you when someone sneezes, or nose-to-nose contact by pigs. However, there are also indirect means of transmission. This would be like if someone at the front of an airplane is sick, and you in the back of the airplane have airborne contaminants or viral particles that are then transmitted. Other common indirect vectors of transmission can include things like ticks, such as the case of African swine fever virus, employees' shoes, vehicles, or in this case, as is the subject of this presentation, feed can be an indirect vector of transmission of viruses. This transmission then requires a host, the individual susceptible to the specific agent, which again can include those same humans or animals. As we consider the role of feed to be an indirect source of transmission of viruses, I'm frequently asked, why are pork producers so worried about viruses in feed? And my thoughts immediately go to porcine epidemic diarrhea virus. This was a terrible virus that was first found in the United States in 2013. Along the y-axis, you can see new cases of PEDV that had been reported, and along the x-axis, we have time from May of 2013 through the current time period in September of 2018. You can see that in May and then again in the following winter, especially in January through March of 2014, we had a huge increase in the number of PEDV cases throughout the United States. This was a very economically troubling and emotionally devastating time for United States pork producers because the virus, porcine epidemic diarrhea virus, causes nearly 100% mortality in suckling pigs and severe morbidity in nursery pigs, as well as some in growing and finishing pigs. As we learn to better control PEDV within the United States, we've still not eliminated it completely. For example, we still have small resurgences of the disease every winter above the red line, which is the epidemic threshold, and so we still have pork producers today struggling with this disease. As we consider how did this virus get into the United States, we've identified that the United States strain of porcine epidemic diarrhea virus is very similar to the strain that was circulating in China at the time. The United States Department of Agriculture conducted a study to best understand the most likely source of entry of PEDV. They determined that the use of flexible intermediate bulk containers, frequently referred to as tote bags, best fit the criteria established for entry into the United States, rapid and widespread across the country, and introduction onto individual farms. In addition to the use of feed transportation methods, we also know that the ingredients or feed itself has caused infectivity. For example, in Canada, there's a research paper that's described the investigation into the role of potentially contaminated feed as a source of the first detected outbreaks of porcine epidemic diarrhea in Canada. In this case, the researchers identified that spray-dried porcine plasma, which had been collected from a farm, was capable of causing infection. Feed that was manufactured from the plasma did not cause infectivity. Likewise, there was an evaluation of contaminated complete feed in the United States as a vehicle for porcine epidemic diarrhea virus infection of naive pigs following consumption through natural feeding behavior in a proof of concept. In this experiment, the researchers collected dust from the inside of feed bins in the United States that had been linked to infection on that farm. They then experimentally fed that to animals and did cause infectivity. Collectively, the role of feed and ingredients led the Canadian Feed Inspection Authority to determine that there was a weight of evidence that linked feed to an outbreak of porcine epidemic diarrhea in the Canadian swine herds. As we look at PEDV, certainly feed, ingredients, and the transportation of feed played a role in either its entry, but certainly its transmission throughout the United States and Canada. Keeping those lessons from PEDV in mind, we now turn to our current concern, African swine fever virus. While PEDV was highly problematic in suckling pigs, there was limited mortality among our grow-finish pig population. That's very different with African swine fever virus, where some of the worst strains have nearly 100% mortality through hemorrhagic fever in the domestic pig population. There is no vaccine and no known treatment for ASF. Fortunately, it is a non-zoonotic disease that does not impact the safety of pork for humans. It is very low transmissibility, meaning that it takes a lot of virus to make an animal sick. However, the problem, particularly as we look at feed as a potential vector, is its high survivability. The virus is very durable, and it can survive in a wide variety of environments and pHs. For example, blood found on a wooden board in one of these countries with circulating African swine fever virus was found to be infectious within still 70 days after it had initially been deposited. African swine fever virus is mainly transmitted by asymptomatic ticks and wild boars. However, other vectors can include domesticated pigs, undercooked pork, vehicles, and, of course, feed. An example from Romania I'll share with you is from the director of a very large pork operation. She told a state news agency, "On Friday morning, I sent samples to the National Reference Lab and tests confirmed the existence of the virus. From Monday, all pigs at the farms, or 140,000, were culled." They stated that the nearby Danube River was a potential source of this transmission. They had heard reports of some smallholders dumping corpses of infected pigs into the Danube River, suggesting that the highly contagious virus might have been spread by river water. There are two cases that I'd like to particularly highlight. The first is the African swine fever virus epidemic in China. Currently, there are 22 outbreaks in 7 provinces that have been reported within the past 25 days. And in just a 21-day period of time between its initial report and 3 weeks later, the virus had traveled 2,100 kilometers. This is concerning because just about half of the world's pigs are in China, and they're almost all exclusively located along the eastern 1/3. The western 2/3 of China is largely mountainous and has some backyard pig populations, but most of the domesticated pig population is held within the eastern 1/3. As of September 11, 2018, China had banned transport of live hogs and pig products from 10 regions bordering the 6 outbreak provinces, and there had been about 30% of China's pigs that had been sent to slaughter. On September 13th, China banned the use of pig blood as a feed ingredient and required all feed that was intended for pigs to be tested for African swine fever virus and found positive to be recalled. Even with these measures in place, on September 17th, 2 new outbreaks were reported in Inner Mongolia. This is troublesome alone, but China's not our only trading partner that has circulating African swine fever virus. We also look to the EU and to Western and Eastern Europe, which is an important trade partner for the United States and has had African swine fever virus circulating for many years. However, it had mainly stayed in Eastern Europe and Russia. However, on September 13th, African swine fever virus was first confirmed in Belgium. This is particularly concerning because at that point, it was 20 kilometers from France and only 65 kilometers from Germany. This caused two major pork producers within Belgium to put a temporary stop on production, and pork exports to the EU and non-EU countries were stopped to avoid recalls. Because of the potential role of vehicle transmission, Belgium actually restricted all automobile access to 63,000 hectares within the country. Currently, they have ASF infected and buffer zones that are created for 24 months. No pigs can be transported in or out of these zones. Any pig with clinical signs requires veterinary consultation within these zones, and all pigs must be kept indoors. Unfortunately, African swine fever virus is not our only concern. Even just within the past week, we've had new reports of foot and mouth disease within China. Japan has reported its first case of classical swine fever virus, and that means that if we're looking for trade partners to bring in feed or ingredients from, we have very limited numbers of countries that are free from foot and mouth disease, classical swine fever virus, and African swine fever virus. In fact, we only have 24, including the United States. But with the break of Belgium last week, we're now down to 23. The simple truth is, we must keep these viruses out of the United States and its feed supply. In order to make that happen, it will take all of us: pork producers, feed manufacturers, and ingredient suppliers. Certainly, feed and ingredients are not the most likely source of transmission. We know that direct transmission is the most likely source of African swine fever virus, foot and mouth disease, and classical swine fever virus transmission. However, feed certainly has played a role in viral transmission in the past, and certainly has the potential to do so in the future. As we consider what we can do to address feed safety, we must recognize that it's a complicated question. In fact, there's a cascade of questions and a cascade of problems that must be circumvented in order to truly prevent any of these viruses from being transmitted through feed. The first question is the ingredient likely to get infected? If it's likely to get infected, can the virus survive transport from the country where it was infected to the point that it would be fed to an animal? If so, is that virus in a form that can cause infectivity above a specific dose that would cause illness within the animal? And if all of those are true, then how can we prevent this? What are things that we can be doing within the United States and abroad to prevent these from occurring? And finally, we need to be prepared in case this occurs. And what can we do to further mitigate any of these options if they cannot be prevented? As we consider this cascade of questions, the first is the ingredient likely to get infected? So what ingredients do you utilize that are at risk for getting infected with the pathogen of concern, whether that be African swine fever virus, classical swine fever virus, or foot and mouth disease? There are a number of factors that come into play. For example, geographical considerations. If the ingredient is from a country or region with an active disease outbreak, particularly if the ingredient is located close to where pigs are breaking with that disease, we would have a higher risk activity. Likewise, agricultural practices come into play. If the ingredient is dried in a way or transported in a way that it is likely to come into contact with wild boars or with fecal contamination, that would be more likely to be infected with one of these foreign animal diseases. And finally, packaging plays a role. For example, those that are transported in single-use bags or totes would be less likely to get infected than those that would be packaged in reused totes or bulk trailers. We also have to consider where are these ingredients from. Along the left is a table of the most commonly imported ingredients according to the U.S. Harmonized Tariff Schedule and reported by D and others in 2016. At that point, a large quantity of soybeans and synthetic lysine and soybean meal was being imported into the United States. Another way of looking at this is who are we importing from? And along the right side of this screen are the top 30 United States United States agricultural import sources from 2013 to 2015. As we look, currently Canada, Mexico, and Australia are free of African swine fever virus, classical swine fever virus, and foot and mouth disease. Only one country in South America is free of all three of these diseases, and certainly as we look at Europe and Southeast Asia, many of our important trading partners are positive with at least one of these circulating diseases. So again, as we look at which ingredients are at risk for becoming infected with a foreign animal disease, we have some examples to consider. A higher risk ingredient would be something like rice hulls or corn cob carriers from a country with circulating African swine fever virus, classical swine fever virus, or foot and mouth disease. Those countries would have the geographical considerations as well as the agricultural practice where rice and corn is frequently dried on roadways and exposed to both wild animal populations and fecal contamination. And finally, those are ingredients that are more likely to be transported in reused totes or bulk containers prior to their arrival at a different type of facility. Meanwhile, a lower risk ingredient would be something like synthetic amino acids that are fermented in a biosecure facility in the same countries, but packaged into individual single-use bags. In this case, even though we're in a country that's high risk, the agricultural practice of how to make synthetic amino acids and how they are packaged reduces the likelihood of being infected to the point that it would be considered lower risk than something like a carrier with rice hulls or corn cobs. If an ingredient is infected, it then has to survive transboundary transport to the United States before it can cause infectivity within the domesticated pig population. That said, we have some research that helps us understand the survivability of viruses in feed and ingredients. The best paper for this subject is published by D and others in 2018, which is titled The Survival of Viral Pathogens in Animal Feed Ingredients under Transboundary Shipping Models. In this case, the researchers took the most frequently imported materials, which are in this first column, including conventional soybean meal, organic soybean meal, soy oil cake, distillers grains, lysine, choline, vitamin D, cat and dog food, and pork sausage casings. And they subjected these ingredients to porcine epidemic diarrhea virus, Seneca virus, which is a surrogate for foot and mouth disease, African swine fever virus, porcine sapal virus, PCV2, pseudorabies virus through a surrogate, and PRRS virus. Any of the cells that are red demonstrate that there was still viable virus at the end of transboundary transport, meaning that in this case the authors infected the soybean meal, in this case conventional soybean meal, with PEDV or any of these other diseases, and then subjected them to similar temperatures and humidities that had been reported during a 37-day transport from Beijing to Des Moines as part of a vessel transport. If there was still viable virus at the end of this 37-day transport, they reported that as red or still potentially infectious. So the red cells represent those that were still capable of surviving this transboundary transport, at least theoretically based on this model. There are a few ways that you can look at this table. First would be by virus type. You can see with PEDV that there are a number of viruses not tested. However, those that were demonstrate that PEDV is highly likely to reside within feed or ingredients. Likewise, Seneca virus, the surrogate for foot and mouth disease, as well as African swine fever virus and porcine sapal virus, survived in nearly all the tested ingredients. Alternatively, PCV2, the surrogate for pseudorabies virus, and PRRS had limited survivability. The other way that you can look at this table is by going across in rows by looking at specific feed ingredients. In this case, you can see that conventional soybean meal was one of the ingredients that harbored these viruses the best, where it was positive for nearly all of the tested viruses, the exception being PCV2. Alternatively, something like distillers grains was less likely to be was less likely to be positive. And in this case, only Seneca virus A, or the foot and mouth disease surrogate, was positive in dried distiller's grains. Another important research paper to help us understand what types of viruses are capable of surviving transboundary transport from point of infection to the United States would be another paper published by D and others, but in 2016. This research only looked at porcine epidemic diarrhea virus, but it was also the same 37-day transboundary environmental conditions. What I want to point out in this case was how organic soybeans and conventional soybeans themselves were not infectious upon arrival. However, the meals, both organic soybean meal and conventional soybean meal, were considered infectious. Another important experiment to consider was first reported by Cochrane and others in 2016 and demonstrates the survivability of viruses in porcine-based ingredients. In this case, the authors artificially inoculated porcine epidemic diarrhea virus into a diet free of porcine-based ingredients, avian blood meal, porcine meat and bone meal, or spray-dried porcine plasma. They then measured the viable quantity of PEDV over time up to 42 days. The quantity of detectable porcine epidemic diarrhea virus in the porcine-based ingredients, both the meat and bone meal and spray-dried porcine plasma, was very stable over the 42 days. However, the quantity of detectable PEDV RNA over time was reduced in the complete diet that had no porcine-based ingredients, and also was substantially reduced in the avian blood meal. While those three research papers together help us understand that there is certainly a pathogen by ingredient, by environmental interaction, we also have to understand that we have very limited data on understanding the survivability of viruses in various ingredients. For example, in this table, I have taken common ingredients in swine feed and placed them in the first column. The next columns represent our knowledge of the ability for foot and mouth disease, classical swine fever virus, African swine fever virus, pseudorabies virus, or porcine epidemic diarrhea virus to survive for at least 37 days in those ingredients. Anything that's in red has research that has demonstrated the ability for that virus to at least theoretically survive under experimental conditions. Anything in green has been tested, but we have not found viable virus after 37 days. And the scary part for me, anything that's in white, has not been evaluated or reported in research. That means that while we have some knowledge that helps us understand that certainly foot and mouth disease, African swine fever virus, and porcine epidemic diarrhea virus can be transmitted through feed or ingredients and survive transboundary transport, there's a lot that we don't know. Based on the limited research that we have available, ingredients that tend to harbor virus include soybean meal, porcine-based ingredients, vitamin premix, choline chloride, and synthetic lysine and synthetic methionine. However, unfortunately, there are still a large number of ingredients that have not been tested for their ability to harbor virus. To help make assertions about ingredients that have not yet been evaluated, it seems that ingredients are more likely to promote virus if they have any of these specific characteristics. Those that are high in protein seem to be more capable of harboring virus, especially those that are high in natural protein. Porcine-based ingredients certainly seem to have the ability to hold on and retain viable virus like spray-dried porcine plasma or porcine meat and bone meal, and this certainly has been reported in African swine fever virus as well. It seems that ingredients with a relatively large surface area relative to mass are more capable of harboring virus. For example, soybean meal has been reported to harbor more virus or be more likely to harbor viable virus than whole intact soybeans, which potentially makes sense. As you see in the scanning electron microscope pictures on the right, you can see that whole soybeans are a relatively smooth surface that have a hull, in many cases, covering any of their exposed proteins. Alternatively, soybean meal has been processed and has certainly lots of nooks and crannies for virus to hide in, but also likely more free amino acid residues and natural proteins that are more likely to bind to or harbor virus. This exposure to protein may be an important factor, but has not yet been confirmed. Finally, microingredients with carriers tend to be more likely to harbor virus than microingredients that don't have carriers. If an ingredient is contaminated with a foreign animal disease and it survives transport to the United States, the next question is if the virus is still infectious upon arrival. This means that the virus must still be in an infectious form, and there must be sufficient quantity of that virus to cause infectivity above a minimum infectious dose. We know the minimum infectious dose of PEDV on day 0. That was reported by Schumacher and others in 2016 and reported to be very low. It's 10 to the 1 TCID50. That's a very low infectious dose. Not very many cells or particles of virus are necessary to cause disease. The minimum infectious dose of African swine fever virus has been reported by a number of others in various strains of the pathogen. However, it has not yet been reported in the highly virulent Georgia strain that's been transmitted through China currently, and it has not been reported through natural consumption behaviors through feed or water consumption. One of the reasons that we have such limited data on infectivity is that our current diagnostic capabilities to assess infectivity are limited, particularly assessing infectivity in feed. The case study that I'll be demonstrating is porcine epidemic diarrhea virus. Here's a virus that's been in the United States for well over 5 years, and our diagnostic capabilities have come light years during that time. However, even today, 5 years later, we predominantly use PCR as a way of demonstrating or measuring porcine epidemic diarrhea virus within feed or ingredients. PCR's advantages is that it's relatively low cost and highly available. Most diagnostic laboratories today have a PEDV-based PCR that's effective to measure the quantity of detectable PEDV within feed or ingredients. Unfortunately, a PCR positive does not necessarily mean that we have infectious virus. All that it means is that there's detectable virus, or portions of the RNA are present and detectable. Furthermore, PCR negative results do not necessarily mean that there's no viable virus present. For example, in the early days of measuring PEDV, many facilities had their cycle threshold set at 35. We subsequently demonstrated that a CT of 37 was capable of detecting of causing infectivity, meaning that you could have a feed sample that tested negative but was still capable of causing infectivity within a bioassay model. Another option for diagnostic capabilities is virus isolation. This is not a widely available or proven method, and a VI negative does not necessarily mean that it was non-infectious. Finally, the gold standard would be a bioassay, where you're actually feeding this material to pigs to understand and evaluate if they get sick with the disease. As you can imagine, bioassays are very expensive and not widely available. And again, a bioassay negative does not necessarily mean that the material was non-infectious. It simply means you were not capable of detecting the infection with the limited number of animals that were being fed the substance. In summary, diagnostic tests have limited capability today, even in PEDV, and they have not been developed or validated for most pathogens and feed or ingredients in the United States. For example, today, none of our diagnostic labs can currently test for African swine fever virus, classical swine fever virus, or foot and mouth disease in feed or ingredient vectors. So as we look at this, we now understand that there is, although limited, sufficient data to help us understand that ingredients can be contaminated with foreign animal diseases. They can potentially survive transport from the countries at risk for infection to the United States. And as far as we know, they can cause infectivity after that shipment. Based on that, it is relevant for us to consider how can we prevent viral transmission through feed or ingredients. One of my larger and overarching concerns is, will we even know if or when a foreign animal disease enters the United States? For example, in the United States, we have found reported porcine epidemic diarrhea virus titers and spray-dried porcine plasma months prior to the initial outbreaks in 2013. That means that we must have had circulating virus if it was found in the spray-dried plasma. Secondly, there are reports that China may have had circulating African swine fever virus since June, or maybe even as early as April, but it was not reported to the OIE until August 3rd. This, combined with the fact that African swine fever virus has an incubation period of about 15 days, means that it's possible for virus to come into the country and begin being transmitted from one location to another before animals even begin seeing disease. And so based on that, it's important and relevant to begin our efforts to ramp up biosecurity to prevent foreign animal disease transmission through feed or ingredients. As we consider this, we will turn to a decision tree that was published by the Swine Health Information Center. This decision tree helps us make decisions about specific feed or ingredients that are from various countries and helps us determine if they are low risk, a minor risk, a moderate or heightened risk. As we look at these different ingredients, we know that those from the countries that have circulating disease would have higher risk than those countries that do not have circulating disease. If there is an ingredient that is likely to be infected through geographical, agricultural, and transportation practices, and that ingredient has demonstrated an ability to harbor virus that is capable of causing infection, such as soybean meal or porcine-based proteins, consider eliminating those high-risk ingredients from the mill and sourcing them from countries that do not have circulating virus. If you're going to eliminate high-risk ingredients, communicate this with procurement. Certainly, we've seen previous instances where there's a decision to exclude a high-risk ingredient from the mill, but procurement has not been informed of this and continues to purchase ingredients. Likewise, confirm that other ingredients are not potentially contaminated through shared lines or transport, and look for non-obvious sources. We've seen producers in the past exclude high-risk ingredients only to find that that exact ingredient ended up being a carrier or blended in to their premix, base mixes, or add packs, and later on cause infectivity. It's also important to only use approved vendors and haulers. Have an understanding of where your ingredient came from. Along the way, did it get blended or mixed with other ingredients? What are the biosecurity policies at each of those locations? These conversations are important to have in order to properly assess ingredient risk for carrying foreign animal diseases. A biosecurity audit is something that is frequently done throughout various types of industries, but has not yet been adapted within or adopted largely within the swine industry. We have an example posted on www.ksuswine.org to help facilities have conversations about potential actions they can take to improve biosecurity. If you ask your supplier to increase biosecurity or exclude some ingredients, understand that it will most likely be associated with a higher cost. At your facility, be diligent with feed mill biosecurity. For example, limit contamination from trucks and people. Remember that African swine fever virus has about a 15-day incubation period. During that time, we certainly could see transmission from trucks and people. To limit that, use receiving mats and funnels to cover the receiving pit so that you limit any foreign material or dirt that can drip into the receiving pit and contaminate your ingredients and subsequently your feed. If possible, don't let drivers out of trucks and instead use your own employees to unload trucks. Mostly, start treating your mill like a farm. Create physical barriers so that truck drivers and other individuals don't go into the main manufacturing areas. Consider using footbaths and employee zoning so that individuals stay in designated areas. In high-stress times, we may need to consider sanitizing trucks. This is something that is relatively common in the broiler breeder industry to protect parent flocks, as well as internationally where there's more pressure of viral transmission through feed or ingredients, or particularly the trucks that are delivering feed or ingredients. Many times we would see people having designated shoes and designated zoning within feed mills, where within the United States, it's still relatively common for a feed mill manager to wear the same boots to work and walk across receiving grates that he may use to feed his pigs at home or walk through the county fair on the weekend. To help understand the importance of keeping virus out of feed mills, we've demonstrated in one research project what occurs within a feed mill for how widely spread PEDV, or porcine epidemic diarrhea virus, can go once it's introduced to the manufacturing environment. In this experiment, we took environmental swabs at baseline before we started mixing any feed, and then we manufactured a PEDV negative diet. Next, we manufactured a diet that included PEDV, so we intentionally inoculated a diet to include this virus. This was done in our Biosafety Level 2 OH Cruise Feed Technology Innovation Center, which is a biocontainment facility that's 3 stories tall, but it still contains common feed manufacturing equipment like mixers, bucket elevators, and pellet mills. So we introduced porcine epidemic diarrhea virus to the manufacturing environment, and then we manufactured 4 subsequent diets that did not contain PEDV. After each one of these time points, we collected environmental samples. The first set of samples we collected were the direct feed contact surfaces, meaning the inside of the conveyors and the inside of the mixers. At baseline and after the negative diet, of course, there was no PEDV detected. However, as soon as we introduced PEDV into the diet, every surface that we tested was PEDV positive, and it retained its positive detectable virus after the first diet was manufactured. After the second, third, and fourth diet, we still had a high level of surfaces, or a large number of surfaces, that were contaminated with detectable PEDV. In addition to the direct feed contact surfaces, we also took environmental samples of adjacent surfaces, such as those that were nearby but not actually touching the feed. So this would be the outside of conveyors or mixers, the floors and walls nearby where we were doing these activities that had dust that we could see were landing on that surface. Again, after baseline and after the negative diet, all of those surfaces were negative. But as soon as we introduced porcine epidemic diarrhea virus to the manufacturing environment, all of those adjacent surfaces went positive, even though we never directly applied the virus to those surfaces. Not only did they go positive, but they retained their positive status throughout the entire manufacturing process. Perhaps most surprising to me is this green line. We also tested environmental surfaces that were relatively far away from the manufacturing surfaces, meaning 2 or 3 floors or several yards away where we could not see dust landing on these surfaces. Again, they were negative after baseline and after the negative diet, but after we introduced porcine epidemic diarrhea virus to the environment, almost all of those surfaces had detectable virus on them. And even after manufacturing 4 subsequent diets, we were unable to remove that residue, and it retained its detectable status with porcine epidemic diarrhea virus over time. This demonstrates an important concept. We need to keep virus out of feed mills. As soon as it's introduced to a feed mill, the dust within that mill is capable of carrying the virus to almost every surface. We subsequently collected samples and dust samples off of some of these environmental surfaces and fed it to pigs, and we were capable of demonstrating infectivity, meaning that not only is this dust causing or harboring virus and spreading virus throughout the feed mill, the dust is also potentially infectious. To help us understand how we can better prevent virus introduction into the feed mill, I think it's important for our industry to consider environmental monitoring. The concept of environmental monitoring is really based on our knowledge from the human and pet food manufacturing areas, where environmental monitoring is used to give them a more consistent and accurate picture of contamination of equipment, ingredients, and finished product. In this case, I have an example of an empty screw conveyor. Yes, that's right. This is an empty screw conveyor. The flighting in that screw conveyor can't touch the housing of it, and so there's a small quantity of feed that has to reside in the bottom of that conveyor between batches. We've known this for some time. This is commonly accepted, and that's why when we have something like a drug or a chemical or nutrient deficiency or toxicity that we're concerned about, we will flush or sequence to get rid of any of this low-level residue that might be stuck in the bottom of conveyors and cause batch-to-batch contamination. Consider, however, if the pellets in the bottom of this screw conveyor contained PEDV. That empty feed conveyor that still has some level of feed would contact the next subsequent feed batch. However, the dust that's on the top of that conveyor would contact multiple subsequent batches. And likewise, the dust is potentially a better indicator of whether virus was ever actually in the conveyor, not necessarily which feed samples may be positive or negative. Maybe a better explanation of this is if we think about it in a tote of an ingredient. Frequently, when we think about contamination, we consider it or assume that it's widely and evenly distributed. However, it's much more likely that something like a virus would actually be isolated in a hot spot in an ingredient. Whether it's a hot spot of an infected tick in that area or fecal contamination, it's highly likely that an affected ingredient or a high-risk ingredient, if infected, that the infectivity would actually occur in a hot spot and not be evenly distributed throughout the material. This makes understanding and trying to find that hot spot and determining the safety of the ingredient particularly challenging. For example, if this tote bag were to arrive at most facilities, we would collect a product sample. And at that point, we could, if we had the diagnostic capabilities, run this sample and try to determine or find any infectious virus. In many cases, based on where that sample is collected, we would assume and it would test safe. Even in the most highly scrutinous sampling procedures, we would sample this tote through 10 different product or probes. Even by probing this ingredient tote 10 different times, it's still possible that we would not actually collect a product sample that was capable of finding any of the infected material. Instead, what we propose is the idea of collecting an environmental sample. By taking a sponge or a swab and swabbing the entire surface of the tote, particularly the top of the tote, you're able to catch the dust that was accumulated and gathered during the loading process. Now you're going to test this to see if it's positive or negative, and it's not nearly going to be as indicative of the level of contamination as if we were to breach that hot spot in the ingredient. However, you should be capable of detecting some low-level contamination and be able to get a broader picture of not necessarily the level, but if that ingredient was actually infected. To give an example of this, one of the things that we've been considering and working on over the past year is understanding how we can better understand breaches in biosecurity and opportunities for improving biosecurity in United States swine feed mills. In this case, we've been sampling 12 different swine feed mills in the United States from North Carolina to Colorado and from Oklahoma to Minnesota. And we've been sampling them over 3 different time periods in the fall, winter, and summer, and determining their overall contamination rate. In this case, we're actually measuring Enterobacteriaceae, or commonly termed EBAC, which is commonly used as an indicator of overall hygiene in the rendering industry, the pet food industry, and the human food industry. What we found is that finished feed has an overall EBAC concentration of about 60%. Now, this alone is not necessarily concerning. EBAC, again, is an indicator of overall hygiene, and it includes pathogenic bacteria. It's a family of bacteria, and so it includes within its family Salmonella and E. coli, but it also includes non-pathogenic bacteria. And so there's not necessarily a gold standard of what level is best, but it does help us understand where is this contamination coming from. Well, if it's in the finished feed, it makes sense that the loadout auger has a similar level of EBAC. The finished feed bin actually has a higher level of EBAC within it, which, based on how feed flows, the frequency of cleaning, and the high moisture levels that can occur in finished feed bins through humidity, it's maybe not altogether surprising that it's a great area to find bacteria. However, we've been trying to figure out if it's in the finished feed bin and in the finished feed, where does it enter? Where is some of this bacteria, or really what are some potential for virus entry within different types of feed systems? One of the concerns was potentially through cooler air. And so in the pellet mill air intake, we actually find a relatively low level of contamination, as we do with the fat intake outlet in the fat intake inlet. What was surprising was the very high level of contamination on ingredient pit grading. Again, this is commonly something that I see having tires rolled over, people's feet walk over this on a regular basis, and certainly the ingredients would go through it. And then floor dust. We see floor dust in receiving, floor dust in manufacturing in the control room and in the warehouse having a relatively high level of contamination, as well as on the broom, and perhaps most surprising is worker shoes. As we look at the most likely sources of bacteria entry into the feed system, worker shoes and the ingredient pit, as well as floor dust, seem to consistently provide the highest levels of entry of bacteria into our feed. Likewise, I think that we can use EBAC as an indicator within swine feed mills to help give us a better understanding of what are our risk points for biosecurity compliance. We've actually demonstrated this in one of these feed mills. So if we take out of this set of 12 different feed mills, here is a single United States swine feed mill that we've been monitoring over time. And we looked at, as we pulled just their data out, very similarly, their ingredient pits, some floor dust, and in particular, the floor dust in receiving, the floor dust in the warehouse, and worker shoes were highlighted as problematic areas that needed to be cleaned up. Within a couple of months of us completing our swabbing experiment with EBAC, unfortunately there was a facility that had been part of our system that broke and was associated with a break in Delta coronavirus. We went back to the facility and collected the same environmental swabs but submitted them for Delta coronavirus and Seneca virus detection. In this case, I've actually overlaid the Enterobacteriaceae contamination rate along the y-axis and then colored the different bars and put the CT values with the Delta coronavirus levels. I know that it's a busy graph, but what I want you to see is in orange would be those that had positive or detectable Delta coronavirus. Those locations that had high levels of bacteria, like the ingredient pit and floor dust in the receiving and in the warehouse, as well as worker shoes, also had Delta coronavirus on them that subsequently was potentially transmitted to a sow farm. The worker shoes are also shown in red because not only did they have Delta coronavirus, but also Seneca virus, which was associated with a separate break with a sow system that was being fed through this feed mill. And so in general, I want us to start thinking about using some type of indicator, whether we're looking for viruses themselves or looking at an indirect indicator like EBAC to help us assess weak points in biosecurity so that we can improve our cleanliness and hygiene of ingredient pits or floors or worker shoes before we have breaks of viruses, either domestic viruses or foreign animal disease viruses through our feed mills. Environmental monitoring is really one that we're considering as a potential option for helping facilities find weak points in their biosecurity. We consistently find risk entry points in worker shoes, floor dust, and receiving pits. If this is something that you'd like to explore, we've posted our methods for how we've conducted this on our website at www.ksuswine.org. So what happens if this enters? What are our options if viruses are being transmitted or have the potential to be transmitted through feeder ingredients? There are a variety of different things we can try, and they can be roughly broken down into point in time versus residual mitigation technologies. Those that are point in time, meaning that we're taking a specific point during the manufacturing practice, and at that specific time, we're going to try to do something to that feed or ingredient to make it safer. These can include things like diluting the virus to below a minimum infectious dose, storing it for an extended period of time, irradiating it, or thermally processing it. As we consider the point-in-time mitigation, I want to again bring up this same research that we had previously described, where we artificially inoculated a feed with porcine epidemic diarrhea virus and watched it spread throughout our feed mill. We took samples at baseline, after the negative diet, after the PEDV diet, and after each of the flushes, and collected those samples and fed them to pigs to assess infectivity. The pigs fed the baseline and negative diet did not have infectivity. However, those fed the diet that was manufactured, after including PEDV, of course, had virus and went bioassay positive. Likewise, the first 2 flushes of ingredients or of feed after the PEDV positive diet was manufactured were also infectious. And so even though PEDV was never intentionally introduced to the diet manufactured first or second after the PEDV diet, they still contained sufficient viral particles to cause infectivity. However, the 3rd and 4th diets were bioassay negative. Again, this doesn't mean that they had no viable virus. In fact, we had detectable virus in these. They just did not have sufficient quantities of detectable virus to cause infectivity within our bioassay model. A follow-up research project to help us better understand the capability of flushes or sequencing to allow us to reduce infectivity was reported by Gebhardt et al. In 2018. In this case, we manufactured a PEDV negative diet, and then we manufactured a PEDV positive diet in the same mixer. After that, we flushed the mixer with a rice hull flush of irradiated rice hulls that had been combined with a 10% inclusion level of medium chain fatty acids, a 1:1:1 inclusion of C6, C8, and C10. After we flushed the mixer with that rice hull flush, we then manufactured a PEDV negative diet, and that's termed subsequent feed. We collected samples in the mixer and in the bucket elevator, and in the negative and positive diet, we fed composite samples where we analyzed samples separately in the mixer and bucket elevator for the flush and subsequent feed. What we found with the cycle threshold of 45 was that, of course, there was no detectable PEDV virus in the negative diet. But when we manufactured a PEDV positive diet, there was detectable virus at about 30.5 CT. Interestingly, there was no detectable virus in the flush of medium chain fatty acids when we took samples at the mixer, but in the flush in the bucket elevator, there was still detectable virus. However, the following feed sample after the flush did not have detectable PEDV according to PCR. We also sent some of these samples to a bioassay, and the negative diet as well as the flush and the feed, were bioassay negative, while the positive diet was, as expected, bioassay positive. This demonstrates the potential, at least, for something like medium-chain fatty acids to be combined with rice hulls to reduce the likelihood of causing infectivity within pigs. I also want to bring your attention to the same diet and slide set that we had previously demonstrated. In this case, we are looking at the inclusion of porcine epidemic diarrhea virus to a porcine-free diet, or porcine-based ingredients from meat and bone meal, spray-dried porcine plasma, or avian blood meal. Certainly, the porcine-free diet and avian blood meal demonstrated that over time the quantity of detectable virus was reduced, but in the porcine-based ingredients, we had a pretty stable level of detectable virus. Irradiation has also been demonstrated to be a highly effective mechanism of reducing detectable quantities of porcine epidemic diarrhea virus, and in this case, virus concentration, as reported by the University of Minnesota. And our team has demonstrated the ability of thermal processing, and in this case pelleting, to reduce the likelihood of infectivity of PEDV. One thing that we need to be careful of, though, is that all of these point-in-time mitigation tactics have the potential for recontamination. In this case, we simply demonstrate that a mash-based diet is inoculated with a specific level of a pathogen. After we pellet, those hot pellet samples are collected and have no viable bacteria in this case. However, after the cooling process, we again have measurable bacteria in them. So where does the bacteria come from? It was frequent within research studies of both bacteria and viruses to find recontamination. And so if we have something like flushing, irradiation, or thermal processing, we have to be careful to protect all of those products after the point-in-time mitigation, because the processing mechanism at that time does not protect them from potential recontamination before the pig actually consumes the product. An alternative is something that has some type of residual protection to it. These would be typically chemical additives like acids or alkalis, essential oils, formaldehyde-based products, or medium-chain fatty acids, where there are products that you could add to the feed or ingredient that would somehow have some type of viability over time to reduce the likelihood of contamination over an extended period of time compared to reducing the likelihood at a single time point. We've done quite a bit of work on this, and specifically within the case of porcine epidemic diarrhea virus. In the black line would be an untreated control, and you can see that at least with porcine epidemic diarrhea virus, things like medium-chain fatty acids have been highly effective at reducing the quantity of detectable virus over time. However, in other ingredients, we've tested a variety of chemical treatments and found very few of them would have capability of substantially reducing the quantity of detectable virus. And so we understand that there's some type of ingredient by time, by chemical treatment interaction, and each of these chemical treatments needs to be evaluated in different types of viruses and different types of ingredients. As we consider the role of transmission to be both feed and ingredients, but also the containers that can carry those feed or ingredients, it's important for us to understand how surface types can also play a role in PEDV transmission. For example, we see that rubber and stainless steel are actually more capable of retaining PEDV than some other types of surfaces, and this makes decontamination of those surfaces particularly challenging. For example, if you were ever in a position where we needed to decontaminate feed mills or manufacturing equipment, we've demonstrated that on steel, straight formaldehyde, concentrated formaldehyde, and concentrated bleach are highly effective at reducing the quantity of porcine epidemic diarrhea virus. However, a number of dry and even other liquid products are particularly problematic at removing PEDV. One of the most concerning things from my side is if we have a virus enter a feed mill, not only do we know that it goes everywhere, but it's very hard to clean. We have tried to clean our biosafety level 2 feed mill just with physical cleaning methods, meaning that we are running through flushing or sequencing, or we are physically cleaning by removing all organic material and all dust that we can through air wands or compressed air. In this case, all that the air does and physical cleaning does is serve to actually increase the overall contamination rate as it spreads virus even more effectively throughout the mill. In fact, what we found is that with every virus that we've tested, in order to get our feed mill clean, we need to tear apart every part of the equipment, all conveyors, bucket elevators, mixers, and we need to power wash it. Power washing it with liquid, and not just straight water, but Synergize, which is a glutaraldehyde blend, and even then we still had detectable virus. And so then we had to move to a 10% bleach solution. And we followed that with a 48-hour heat cycle where the facility was held at 140 degrees Fahrenheit. So based on our data with porcine epidemic diarrhea virus, which is actually less stable than something like African swine fever virus, in our facility to get it clean, we had to tear apart every conveyor and pressure wash it with a 10% bleach blend. If that's not a reason to make sure we keep viruses out of a feed mill, I don't know what is. So in summary, one of the things that we need to think about is that point-in-time mitigants, like irradiation, quarantine time, as well as thermal processing, require the prevention of post-processing cross-contamination. They occur and the protection occurs at one specific point. And so it's important for us to protect those products from subsequent cross-contamination, which is hard to do, as we've demonstrated, because as soon as we introduce virus to an environment, it tends to go everywhere. Secondly, the efficacy of chemical additives, if we're trying to add something that would have some type of residual mitigation, depends upon the type of virus or bacteria. It certainly depends on the ingredient or the feed, and these things fluctuate and vary over time. Unfortunately, the efficacy of both point-in-time mitigants and chemical additives are typically unknown in almost all of the foreign animal viruses that we're concerned with. And so here we come an hour in to potential mitigation strategies. Within the past month, we've been asked frequently for options for potentially mitigating African swine fever virus, classical swine fever virus, and foot and mouth disease in feed and ingredients. And we've heard about quarantine time, we've been asked about formaldehyde, and we've been asked about medium chain fatty acids or other acids that have been demonstrated to have efficacy within PEDV. First, it's important to point out that modes of action differ across mitigant type. Here on the left, I've demonstrated and I have the viral structure of PEDV. We think that the spike glycoprotein, these yellow structures that are on the outside of PEDV, play a really important part in the ability for mitigants to be effective within PEDV and it ultimately to contaminate an ingredient and cause infectivity within a pig. That remains unknown within African swine fever virus and classical swine fever virus. However, both have lipid membranes. One of the most common questions that we've received is how long should I quarantine a high-risk ingredient from African swine fever virus or classical swine fever virus positive countries? At the current time, we believe that there is insufficient data to confidently use quarantine time as a mitigation measure. The best research that we have is the transboundary paper published in 2018 by Dee and others mentioned before. And this paper includes half-life estimates of the ingredients that retained viral infectivity after transboundary shipment. Remember, there was no viral activity in classical swine fever virus, so the only published half-lives are for African swine fever virus based on initial or day 0 and endpoint or day 30 or 37 titers. We have to be careful with this data, and we need to apply these estimates with caution because there's not very many replicates, just 2 samples per ingredient. There was a very high level of the inoculum, 10 to the 5th, and there's an extremely small size of the sample, just 5 grams. And probably most concerning is that these values were determined based on just 2 time points. days, and therefore assume a linear decline in African swine fever virus to be able to calculate a quarantine time. However, we've learned through porcine epidemic diarrhea virus that we actually don't have a linear decline. We have a quadratic pattern of viral decline, and that quadratic pattern differs among various feed or ingredients. For example, here on the left, the quantity of detectable PEDV over a 42-day period in 3 ingredients and a complete diet, as reported by Cochrane and others in 2016 and described previously, is shown. This graph shows the initial, or day 0, and final, or day 42, quantity of PEDV. We've known the known minimum infectious dose for PEDV in a complete diet, which is equivalent to a CT of 37 as measured by a real-time qPCR and shown here in the blue box, and that was reported by Schumacher and others in 2016. If we're going to fit a line and use it to estimate quarantine time, our estimate would be that the quantity of detectable PEDV would be reduced to a level beyond the minimum infectious dose, or that 37 CT, after 60 days in the complete diet. And in porcine meat and bone meal, it would be after 185 days. This alone is a risky determination because we know that the minimum infectious dose for feed at day 0 is 37, but we've extended that to not just day 0, but across all day 42. And that was actually measured in a porcine-free diet. It was not measured in porcine meat and bone meal. We don't know the minimum infectious dose of PEDV in a porcine meat and bone meal. In reality, the research included intermediate sampling dates. So this graph on the right shows the quantity of detectable PEDV on day 0, 1, 3, 7, 14, 21, and 42. This helps demonstrate that the PEDV RNA is relatively stable in porcine-based products, but shows a quadratic decline in the complete diet and avian blood meal. When we tried to apply theoretical calculations to extrapolate the data beyond day 42, the lines for the tested product never reached the minimum infectious dose. I think this concept helps demonstrate the potential for inaccurate assumptions when calculating quarantine times based on 2 time points. The viability of African swine fever virus in ingredients after 30 days remains unknown, and we feel that there's insufficient data to accurately measure it. And so until more research is reported, high-risk ingredients should be sourced from ASF and CSF-free countries, because these realistic quarantine recommendations cannot yet be calculated. We've also been asked frequently about the role of formaldehyde-based products and the research that we're aware of to prevent African swine fever virus transmission through feed or ingredients after they've been treated with something like Salcurb or Terminate. There's no available data to make this determination. Its proposed mode of action through irreversible cross-linkages of proteins are likely to affect proteins of ASF and CSF, but this has not been confirmed, and certainly the spike protein of PEDV is very different in structure than the proteins and protein structure in African swine fever virus. In fact, compared to PEDV as we've described, ASF is just much more environmentally stable across a wider pH range. And so that may affect the efficacy of formaldehyde-based products, and so it's not appropriate to just completely expand and assume what was effective in PEDV is also effective in African swine fever virus. That said, according to the OIE, formalin is an effective sanitizer of ASF, showing that formaldehyde-based products may have the potential to be successful mitigants of African swine fever virus in feed, but we do not have research to confirm this yet. And perhaps more importantly, the FDA approval for formaldehyde inclusion in feed is at 37%, and it is only used to maintain complete feed and ingredients salmonella negative for up to 21 days. It is not approved for prevention or mitigation of porcine epidemic diarrhea virus or any other virus, including foreign animal diseases. The modes of action certainly do differ across mitigant type, and here's that structure of PEDV again compared to African swine fever virus. And I pulled this up because we're again frequently asked if medium-chain fatty acid-based products prevent African swine fever virus transmission. We again have no data to make this determination. Our hypothesized mode of action of medium chain fatty acids is that in the lipid bilayer, which is shown here on the bottom, that those medium chains create pores in PEDV membranes that lead to viral instability. And we think that this may be similar to the mechanism in African swine fever virus. As you look at membranes and lipid membranes, African swine fever virus actually has two lipid membranes, where PEDV has one. And so there's potential that medium chain fatty acids may actually be more effective as mitigants in African swine fever virus than PEDV. However, this has not been confirmed, and research is ongoing in this area. So with all of this, we don't know. What can swine producers and feed mills do to prevent African swine fever virus and classical swine fever virus in feed? Well, with these, we've published a recent frequently asked questions about ASF and CSF in feed document that's available on our website at ksuswine.org, and we encourage you to look at this document. It includes recommendations like keeping out high-risk ingredients. It includes these questions about the current research or recommendations for formaldehyde or medium chain or quarantine time to prevent ASF transmission. And so at your facility, please be diligent with feed mill biosecurity. Cover those receiving pits when not in use, effectively control pests, prevent inclusion of dust from dust collection systems that may recycle that dust back into diets, and manage personnel movement throughout the mill. Many of these references on feed mill biosecurity can be found on our website. And then finally, consider introducing environmental monitoring to monitor your environment for biosecurity compliance. Environmental monitoring is routine in human food and pet food industries and can help identify weak points in biosecurity plans. We know a lot more about porcine epidemic diarrhea virus and viral transmission in feed than we did 5 years ago, but unfortunately, we still know very little about foreign animal disease transmission. Point-in-time mitigants help reduce the risk, but cross or recontamination must be prevented, and this is particularly challenging with viruses that tend to go everywhere after they've been introduced into the environment. Therefore, it may be appropriate to consider chemical additives, especially as more data becomes available. We also really recommend that you have a plan in place for mill decontamination. It is not a fun process. Right now, what it involves is physically cleaning all surfaces, including equipment internal components, power washing everything with 10% bleach, and then rinsing and air drying while preventing recontamination. We hope that no facilities will have to go through this because that's an ugly process and feed mills were not designed to do that. And perhaps more than anything, that's why we highly recommend improving biosecurity to try to keep these viruses out of the feed mill. As we conclude, we need to recognize that feed is just one of the many potential vectors of pathogens. Remember, direct contact by nose-to-nose contact is by far the most likely method of viral transfer. However, as we look at potential indirect transmission of viruses from foreign countries and into the United States, we have to recognize the risk for potential transmission through feed. Based on that, we unfortunately have limited information about all 5 steps for how that occurs. We have limited knowledge on the likelihood for ingredients to get infected, or its survival, its infectivity, prevention, and mitigation. However, what we do know is there's sufficient risk that we should be concerned about African swine fever virus transmission into the United States through ingredients. Because of that, exclusion of high-risk ingredients from high-risk countries is currently the best strategy to help keep these foreign animal diseases out of the country. We recommend that you in feed mills and ingredient facilities ramp up biosecurity and extend it from the farm onto the feed mill. Do not reuse totes and bags, as those have been linked to transmission and entry of foreign animal disease in the past. Consider feed and environmental testing as a way of understanding what are your opportunities for improving biosecurity within your facility. And consider the role of proactive mitigants. There's limited research as we sit here today in September of 2018, but certainly there's things that we can do to be proactive and both exclude as well as try to minimize any potential transmission as we move forward. That said, we have to make logical decisions based on data. Stay tuned as more research is being reported. There is current research on the minimum infectious dose of African swine fever virus in feed and in water. There's current research on mitigation in African swine fever virus and classical swine fever virus through the use of formaldehyde-based products and the use of medium-chain fatty acids. And there's current research on understanding the role of environmental monitoring to be a better indicator of viral transmission risk than something like product sampling and probing totes. All of this research will likely be reported here on our website within the next several months. And so please go to ksuswine.org and feel free to drop us emails and ask us questions as you have questions about the risk of viral transmission through swine feed. Thank you.